This is a question about DNA replication, and the question says describe, so a series of steps of how it happens, how DNA replicates. You may use annotated diagrams. So part one is that DNA unwinds. Is a helix, there's a double helix, that double helix needs to unwind, and the strands separate or unzip, and that's using the enzyme helicase. The hydrogen bonds between the bases break. The bases are exposed. So you end up with two strands with the bases exposed. You've then got three which are floating around in the cytoplasm. Three nucleotides. They are complementary to the nucleotides that are on the exposed bases. So the complementary nucleotides bond, and that's using DNA polymerase. So another enzyme. A pairs with T, and C pairs with G. So remember, apples on trees, cars in garages. That joining forms a polynucleotide chain. You've got phosphodiester bonds along the backbone of that chain, so the sugar phosphate bonds, and you started with two strands, all right, making up one strand of DNA, and this is called semi-conservative because after each strand of DNA is made from the original strand and a new strand. So number 12, you can mention that it's conservative. It's a 7 marking point question, and there's 12 marking points there that we've gone through. This is a copy of the mark scheme. You can see there are AVPs up to 2 marks for naming the enzyme like helicase and talking about that the strands are anti-parallel to each other.